Everyone, myself Gajanan. Today we are looking at the red blood cells from Harper's Biochemistry, chapter 53. Let's get started. Then RBC, that is red blood cells. What is an RBC? Also called as erythrocyte. Erythrocyte is a pack of hemoglobin and carbonic anhydrase inside in the cell. It's called as the erythrocyte, in which hemoglobin stores the or transports the oxygen and carbon dioxide to and from the peripheral tissues and the lungs, and the carbonic anhydrase is the enzyme which is responsible for the conversion of carbon dioxide into the carbonic acid and the bicarbonate. Then what is the anemia? The anemia is the term which defines as the deficiency in the level of red blood cells in the blood circulation. That means if the red blood cells are below the 120 grams per liter, it's called as the anemic or anemia, or if it's above the 130, it's called as the polycythemia. Then the anemia can be arise from the genetic abnormalities such as in the sickle cell disease, pernicious anemia, and then excessive bleeding can also cause the anemia. Insufficiency of dietary iron, vitamin B12 absorption or availability of vitamin B12, or the lysis like of the red blood cells, means destruction of the red blood cells also cause the anemias. Then what is the platelet? Platelet is a substance which forms the clotting at the point where we bleed or where we have cut or bruisings. Then this hemorrhage can be reduced by the speed of formation of structural integrity protective clots. This is a short point for the platelets. Then what is thrombocytopenia? Thrombocytopenia is the low platelet count known as the thrombocytopenia. It can be triggered by bacterial infections, medications including sulfa-containing antibiotics, or autoimmune reactions such as idiopathic thrombocytopenias. Then, other pathophysiologic syndromes such as von Willebrand disease or Glanzmann thrombasthenia are von Willebrand disease and the Glanzmann thrombasthenia are the bleeding disorder or hemorrhagic disorder. In both of them, the platelet fails to aggregate or forms adherence or forms the perfect clot for blood stopping from the cut, but their platelet number is not reduced. Then, more about the red blood cells. This is the process of erythropoiesis. In short, before erythropoiesis, we will see erythropoiesis here. Before that, I would like to say that this erythropoiesis starts from the hemocytoblast, that is the RBC forming cell. Not RBC forming only, it can form RBC, WBC, platelets, or even lymphocytes. The RBCs are or WBCs and the platelets are formed from the myeloid progenitor, while the lymphocytes are formed by the lymphoid progenitor. Then, these hemocytoblasts are stem cells. They possess an unique capacity. They have two capacities, that they are self renewal and they have potency. Self renewal means they can produce another hemocytoblast, means they can multiply. And the second quality of there is the potency, means they can produce RBCs, WBCs, platelet, mean various other kinds of cells rather than the another hemocytoblast. Then potency have another types, that is totipotent, unipotent, pluripotent, multipotent. In that, totipotent is the cell which can produce all kind of cells in an organism, while unipotent stem cells can produce only one type of cell. Pluripotent cell can produce any of the three germ layer, that is mesoderm, endoderm, and ectoderm. Then what is a multipotent? Multipotent stem cells is restricted to forming cells that belongs to a single family or a class. Means multipotent cell can form only the cells of a single family or class. Means if stem cell is going to produce renal cell, then it can produce only renal cell. If it's going to produce only gastric cells, it's going to produce only and only gastric cells. In addition, stem cells can be classified as embryonic and adult, which in latter are limited to their capacity to differentiate. I mean, after some time, the adult stem cells cannot differentiate, but the embryonic stem cells are able to differentiate. Okay, then erythropoiesis in short, because we saw erythropoiesis in much more detail in the physiology. Erythropoiesis starts with a hemocytoblast, that is the common cell for both of these groups. This cytoblast is acted by interleukin 1, 3, and 6, and colony stimulating factor and the cell stimulating factor. All of this combines and forms the myeloid progenitor. It is a precursor cell for erythrocyte, platelets, and the WBCs. and then lymphoid progenitor is the uh, common progenitor or the mother cell for the lymphocytes. In that, who determines that this particular myeloid cell will form the RBC, or this particular cell will form the megakaryocyte or the platelets? There are the enzymes secreted by the kidney, that is the erythropoietin and the thrombopoietin. Erythropoietin will divert the myeloid progenitor towards the RBC, while thrombopoietin will divert towards the formation of platelet. This is what is explained in this paragraph. Briefly, I explained it briefly. Then RBC specialized cells are highly specialized. Now, we have to transport, or our peripheral tissues, our body needs much more quantity of oxygen. That's why our RBCs are modified at a such extent they have nothing inside of them except hemoglobin and some enzymes and other things, some other things, but the main organelle is like mitochondria, then nucleus, then lysosomes are not present there. They are enucleated red blood cell. As there is no mitochondria, means there will be no etc cycle, there will be no Krebs cycle or TCA cycle, and that means our RBC is not going to consume the oxygen that the cell is transporting. That means the RBC will be able to transport maximum amount of oxygen to the body. Then this RBC's biconcave shape is maintained by the some protein, that is pectin, ankyrin, actin, then band protein 4.1, 4.2, and some adenosines and many other protein that we'll see later in this chapter only. Then this disc-like configuration possesses then shape function of the RBC. What is the shape function? The first function of the shape of the RBC, that is the biconcave shape. The biconcave shape of RBC gives the more surface area to the volume in the compared to the spherical forms, so that the more number of oxygen can be diffused inside the RBCs and can be transported. Then second, it enables the red blood cell to fold over and squeeze through. Means at some sinusoidal capillaries, the diameter of capillary is way smaller than the diameter or the shape of an RBC. That's why the RBCs have a property that they can, this is an RBC, it can squeeze and go into the capillaries and transport the oxygen. This is one characteristic. This is the second, and this that's it. For the shape function, we have another characteristic also. Then RBCs travels at the 2 millimeter per second speed. Then, as we know, the RBC is divided of all its organelles. Then the question is how the RBC is going to get ATP or the energy for its own function. The answer is glycolysis. Glycolysis is not dependent on oxygen. That's why the ATP, the maximum number of ATP that RBC can produce, is from the glycolysis. Then this glycolysis is conducted with the glycolysis is conducted inside the RBC. The glucose enters the red blood cell through the process of facilitated diffusion, and this process is mediated by the glucose transporter, that is the glucagon transporter, which is also known as the glucose permease. Glucose permease, and this glycolytic pathway in the red blood cell also possesses a unique branch or shunt, and this shunt is known as the Rappaport-Lewbering shunt or Rappaport-Lewbering pathway. The purpose of it is to isomerize 1,3-bis phosphoglycerate to 2,3-bis phosphoglycerate. The purpose of it, what happens in the normal glycolysis is that from the fructose get isomerized and phosphorylated, and then this phosphorylated fructose converts into the 1,3-BPG, and this 1,3-BPG will be then converted into the 3-phosphoglycerate. But in the glycolysis of RBC, what happen is that 1,3-bis phosphoglycerate will be converted to 2,3-bis phosphoglycerate, because this 2,3-bis phosphoglycerate is what it stabilizes the hemoglobin in the T state. In the T state, what is a T state? There are two states of hemoglobin that we saw in chapter 6. One is the T state, mean low oxygen affinity, means the hemoglobin is ready to give out his oxygen to the peripheral tissues, and the R state means the hemoglobin wants oxygen, or hemoglobin is ready to take oxygen bind. That means the R hemoglobin is mainly present in the lungs, while the T hemoglobin is mainly present in the peripheral tissue capillaries or the arteries. Then This conversion of 13-BPG to 23-BPG is mainly catalyzed by the two enzymes. One is the mutase, that is 23-BPG mutase, and another is the multiple inositol pyrophosphate phosphatase. The difference in these both enzyme is only one, that this 2,3-BPG mutase produces byproduct as the 3-phosphoglycerate, while the this multiple inositol polyphosphate phosphatase produces the 2-phosphoglycerate as its byproduct. These all enzymes are sensitive to pH, and this all this information and other information about the RBC glycolysis from other chapters is summarized in this table 53.2. Moving toward the next topic, as the carbonic anhydrase, as I told you in the information or the bio importance page of this chapter, that the carbonic anhydrase is mainly important for the conversion of carbon dioxide into the carbonic acid or the bicarbonate, because the hemoglobin is relatively more have more affinity towards the carbonic anhydrase or the bicarbonate compared to the CO2. That's the only reason why our carbon dioxide get converted into the carbonic acid and the bicarbonate. Then this rapid conversion happens at the peripheral tissue level, and when this bicarbonate or the carbonic acid reaches to the lungs, this process gets totally reversed and converts it again into the CO2, and it will be exhaled out of the lungs. Then 80% of the carbon dioxide is expired or the given out from the body by this carbonic anhydrase or this process by the hydration of the carbon dioxide. But the another 20% is directly, it binds to the directly to the RBCs and forms the hemoglobin bound carbamates and gets transported. Then RBCs must be continuously replaced. As all we know, our body contains at least 20 to 30 trillion RBCs, and 1% of is easily getting replaced every second in our body, and that calculate towards the 2 million RBCs per second. As our body is producing RBCs at a it way faster rate, our bone marrow secretes or gives out RBCs in the form of reticulocytes. These reticulocytes are not completely devoid of all the organelles. It has some amount of ribosome, endoplasmic reticulum, and mitochondria, and mRNA, vestigial mRNA. And with the help of this vestigial mRNA, in the first 24 hours, the reticulocyte produces polypeptides. Example is the globin under the direction of mRNA molecule. That's it. But what happens in the rare cases, genetic mutation can occur, and this genetic mutation of the ribosomes are called as ribomyopathies. In that example, we can say is the diamond blackfan anemia, which is caused by the mutation in the gene RPS19, and the Fayuque syndrome, which is also caused by the mutation in the gene RPS14. This is just for information and to remember, because we are not yet scientists to find out this what's happening inside and all doing all these things. Just remember this information. Then erythropoietin regulate the production of RBCs. As I said earlier, interleukin 1, 3, and 6, CSF, SCF are all acting on the erythropoiesis. But who decides? That the erythropoietin is the one who decides. The myoid cell will become the RBC, and another myoid cell will become the WBC or the platelet. Then this information, there is information about EPO, that is erythropoietin. As I said earlier, that EPO is mainly synthesized by the kidneys and which is released into the bloodstream in the response of hypoxia. The thing is, our kidneys proximal cell contains connected to PCT cell contains the genes which are specifically responsible for the erythropoietin production. These genes get activated and produces erythropoietin once they are exposed to the hypoxia, means low oxygen levels. It can happen in the case of some accident, or if you are traveling some high altitude places, it can happen there. Then this erythropoietin is administered therapeutically sometimes, not sometimes, like whenever there is a kidney failure in a patient, that means the patient cannot produce poietin no longer. Then we have to give erythropoietin from outside so that our bone marrow can form the RBCs at a faster rate, or at least our bone marrow can match the normal rate of our RBC production and can save us from the anemia. Then this pharmaceutical EPO or the erythropoietin is uh, synthesized from the Chinese hamster ovary lines. These are the cells from the ovary lines of the Chinese hamster. Then the next point is oxygen. Oxidation of heme ion compromises oxygen transport. That means if the oxidation happens in the hemoglobin, it our hemoglobin will be less responsible or won't be able to transport oxygen that faster or at that quantity. What happens actually is the our hemoglobin converts to the meth hemoglobin because of the ROS, reactive oxygen species, that we'll see in the next point in this point. Then what is methemoglobin? Methemoglobin is the hemoglobin of the Fe3 plus ion. Normally in the normal structure of hemoglobin we have ferrous form, that is the Fe2 plus form, but because of the ROS species or the reactive oxygen species, our hemoglobin converts to the methemoglobin. This ferric heme do not bind to the oxygen, which reduces the oxygen binding site and less oxygen will be transported. And certainly we can reverse this process, and our body does this absolutely. In red blood cells, methemoglobin reduction is catalyzed by the enzyme NADH cytochrome B5 methanoglobin reductase system, and this system has two components. The first component is the cytochrome b5 reductase, and the second component, this reduced cytochrome b5 will transfer the electron. We'll see. Then this is the complete reaction. What happens is the cytochrome b5 oxidized, this is in the oxidized form. This NADH have an one hydrogen, means one electron is present here. This one electron will be transferred to the oxidized cytochrome b5, and this cytochrome b5 will become the reduced cytochrome b5, and this NAD can again go to the process of glycolysis certainly and gets converted into the NADH and can carry again one electron. This cycle will continue. Then what happens, this is the product of this reaction which we form, this cytochrome b5 reduced. Then this cytochrome b reduced b5 or the reduced cytochrome b5 will transfer its electron to the methemoglobin, that is the hemoglobin with the Fe3, that is the ferritin structure or the ferric structure. This electron will be transported to the methemoglobin, and the methemoglobin will converted into the normal hemoglobin, and this cytochrome or the reduced cytochrome B5 will again be converted into the oxidized B5, and this process will again continue. And that's the reason our blood does not present methemoglobin in the much more quantities in the hematocrit. Hematocrit is the blood sample or the 1 microliter of a blood which we take for the sampling purposes. Then the ultimate source of electron in the reducing of that's what I said. The source of this NADH will go to the glycolysis and will carry electrons. Then the result of this system is the result of this system is the trace quantities of methemoglobin are normally present in the erythrocyte. Okay. Then, what is methemoglobinia or the methemoglobinia? Methemoglobinia is the abnormal accumulation of the methemoglobin inside an RBC, which arises from the genetic abnormalities. There are two types of methemoglobin anemia or the methemoglobinia: inherited and acquired. This inherited methemoglobinia can be from the genetic abnormalities, and the acquired methemoglobinia can be formed by the nitrites, aniline, or the sulfamide antibiotics. Then the affected patient generally presents with the bluish discoloration of the skin and the mucous membrane of the skin, that is the cyanosis we see. It is most commonly arrived from the deficiency on the quantities and activities of cytochrome B5 reductase. Yeah. If this process is not gonna conducted properly, that means we will have less amount of RBCs and more amount of hemoglobin. Less amount of RBCs cause the anemia, and the this destructed hemoglobin forms the methemoglobin and combined is the methemoglobinia, which might have it can be reversed by the cytochrome b5 reductase system, but if it doesn't, it forms this condition. In this, or HBM in this is this is the one which is much more important for us. Other thing you can remember for your fun. HBM Boston, which causes the alpha subunit mutation at the alpha subunit of the hemoglobin, which will be replaced by the tyrosine. And there is one exception of the valine in the case of HBM MILAC 1, where the valine 67 will be replaced by the beta subunit or by the glutamate. It's also known as which heterozygous. It's no, it's okay, fine. Then superoxide dismutase catalase glutathione protect the RBCs from oxidative damage. Certainly, this is the process of oxidation with that we saw. Now we need some antioxidants in our body, that in that super oxide dismutase and the glutathione and catalase acts as the major antioxidants of our bodies, which counters the oxidation process and saves our RBC from the destruction. And that superoxide, that is the O2 in its reduced form, or yeah, reduced form, which is generated from the red blood cell auto oxidation of the hemoglobin to methemoglobin, means when the hemoglobin get converted to the methemoglobin, it forms the one molecule of the molecules of superoxide. There is another source of superoxide also, that is this enzyme, that is NADPH hemoprotein reductase, which also does the same process, that catalyze the reduction of the Fe3 into the Fe3 in methemoglobin to the Fe2 and forms the superoxide species. What does do this ROS? Then ROS can react with the substances like protein, lipids, nucleotides, and other biomolecules, and can damage also, which are vital for our body. Then another effect of the oxidation of the iron or the hemoglobin is that the in our body, this storage of Fe3 or the iron storage known as the ferritin, if this ferritin is get oxidized by the superoxide, which results in the formation of Fe2 plus, that is the good molecule. But while catalyzing this reaction forms the OH free radicals. These free radicals are again harmful for our tissues, which can damage our cells. Then this superoxide may also result in the tissue damage, which I told you already. And then yeah, this superoxide can also affect the individuals which are suffering from the high iron absorption, which can be seen in the condition of hereditary hemochromatosis. Means more iron, and then will convert this more iron from ferric to a ferrous form, and then this conversion will again yield the superoxide bodies. Then it will try to counterpart this process, and then will again form the OH or the OH free radical, which is again harmful for our. In short, the effect of this all reactive oxygen species is that destruction of our cells, main vital proteins, lipids, and nucleotides and biomolecules, and the destructions of the peripheral tissues. That's it. Then hemolytic anemia. There are many other types of hemolytic anemia. In that, the G6PD is the major one. What is an G6PD? If you can remember, G6PD or the glucose 6 phosphate dehydrogenase is the main enzyme in the process of pentose phosphate pathway for the reduction of NADP to the NADPH. This deficiency is most common in the enzymopathies, because this is an enzyme. If the deficiency of this will form the enzymopathies, absolutely. Then more than 400 million people are estimated to carry one. This is of no use, okay, like use, but for fun you can remember. The deficiency is the most common among the tropical African, this is important, Mandarin, and the certain parts of Asia. The inability of to generate more NADPH molecule will result in the oxidation of glutathione, and the glutathione is very very good antioxidant, and it's very important for the counterpart of these superoxide species or the ROS species, so that our hemoglobin can be saved from the oxidation to be converted into the meth hemoglobin. Then what if this glutathione is oxidized? What will happen? This oxidation of glutathione will form the Haynes bodies. The Haynes bodies are the insoluble aggregates of the hemoglobin. The thiol group of hemoglobin, that is the SH group, will be oxidized, which happens in the sickle cell anemia, oxidized like in the sickle cell trait, not in anemia, sorry. And the persistence of this genetic variant has been attributed to the potential to confer enhanced resistance to malaria. Yeah, like for sure, in the sickle cell disease or trait, the patient develop like resistance toward the malaria, but the more harm is that having a sickle shape of an RBC, and the RBC won't be able to carry oxygen and transport the oxygen, and the patient will be anemic again. Then hemolytic anemia can be caused by the three factor: intrinsic, extrinsic, and the membrane specific factor. And that extrinsic factor that are outside of the RBCs will include the hypersplenism. Hypersplenism is the increased and large shape of an spleen, which is we also known as graveyard of RBC. Why? Because this spleen have an sinusoidal capillaries. These sinusoidal capillaries are very small capillaries, and the if RBCs are in a larger shape or the bigger shape and gets trapped in the membrane of this sinusoidal capillaries, the macrophage will destroy them, will digest them, and this is why will our body will exhibit the hemolytic anemia in the form of extrinsic form. Then there are some transfusion reactions: RH disease, warm and cold body hemolytic anemias, and hemolysis. In that transfusion reactions, that means if the person is like administered or receives a transfusion from the another person by the incompatible and another person, which will lies the RBCs of this person, means incorrect transfusion, we can say mismatch transfusion in the case of mismatch transfusion. Then what is an RH disease? RH disease is a specific type of erythroblastosis fetalis, where the Rh positive fetus is carried by the Rh mother, Rh negative mother. What happens is the platelets or the antibodies of the Rh negative mother crosses the placenta and will destroy the platelets of the Rh positive fetus, and the fetus will be the anemic fetus. The consequence of this autoimmune disorder is the warm and cold antibody. This warm and cold antibody hemolytic anemia is can be triggered by the temperature, outside temperature. Like if warm temperature, that means warm hemolytic anemia. Cold means cold. Then some infection toxins and some factors. The infection toxins given by the or the uh, by the snake bites or the snake venoms, some proteases and phospholipases from insects and reptiles, some infectious bacteria can also give us some factors like Escherichia coli and Clostridia can give us some factors which will attack and lyse our red blood cell membranes, and these factors will collectively referred as the hemolysins, which can be composed of proteins, lipids, and combinations of all of them. Then parasites like Plasmodium can cause malaria, and also can cause the hemolytic anemia, means the destruction of RBCs. In short, that's you must know at this point. Then this intrinsic causes we know in the extrinsic, that is hypersplenism, RH disease, transfusion reaction, antibody reactions, warm and cold, then hemolysis. Now the main cause, the intrinsic cause is the G6PD deficiency. We saw it already. Then the second major cause is the hemoglobinopathies. That means the defects in the composition or the structure of hemoglobin. uh, Most hemoglobinopathies are the sickle cell anemia and the various thalassemias, which we already saw in chapter 6, which are genetic in nature, and it's rare that hemolytic anemia can arise from the insufficiency of the enzyme, that is the pyruvate kinase, Pyruvate kinase is the main or the important enzyme where we yield two amounts of or the two molecules of ATP from the pyruvate to the paper to phosphoenol pyruvate to the pyruvate in the process of glycolysis. As there is a defect in this pyruvate kinase or this enzyme is not adequately produced or used, we will have or the RBCs will have less number of ATPs and will fail to export the excess water and sodium outside of the cell, which will result in the building of an osmotic pressure, and the cell will become turgid and will be trapped by again the spleen, and it will cause the destruction by the macrophages in the capillaries, the sinusoidal capillaries of the spleen. Then the third cause of the hemolytic anemia, that is the membrane specific, that causes of hemolytic anemia, that is the membrane specific. What are the membrane specific causes? There's the uh, osmotic pressure buildup by the, like there is a failure in the some proteins like spectrin, ankyrin, actin. These are the membrane proteins. Like membrane specific means the defect in the proteins which maintains the biconcave shape of an RBC. These proteins are spectrin, ankyrin, actin, band 4.1, band 3, band 4.2, band 4.9, adenosine, and some many other proteins which we'll discuss later. In that, the most important of the disease is the or the condition is the hereditary spherocytosis and hereditary elliptocytosis. I'll just give you a brief overview about this. Hereditary spherocytosis is also caused by the genetic mutation in the structure of spectrin. uh, in this condition, the shape of an RBC becomes spherical. It will become more and more globular, and in the case of, it's like obvious, the RBC will become elliptical in shape. Then, what is the paroxysmal nocturnal hemoglobinuria? Paroxysmal nocturnal hemoglobinuria is the presence of hemoglobin in the urine, specifically in the nighttime, specifically in the nighttime, which is caused by the defects in the synthesis of glycophosphatidylenosidal uh, groups such as the acetylcholine esterase, which is useful for the esterification or the destruction of the acetylcholine, and the decay accelerating factor, which is responsible for the maintaining and regulation of the complement system. Complement system is like an not like an it is an immune system which will counter microbes and some pathogens to enter the red blood cell. Failure to do it will cause the paroxysmal nocturnal hemoglobinuria. Then red blood cell and its membrane. This early analysis of sds page. Sds page is a technique where what we do is we perform the electrophoresis and the buffer solution, and we see which protein moves towards its opposite side. Like if, let's say, I'm not sure this about spectrin, let's say the spectrin is the positively charged protein. What will happen in the electrophoresis? This spectrin will move to the negatively charged pole or the negatively charged electrode, and we will calculate the speed of this movement of these proteins and will note down. In that, the molecule which moves the slowest will be considered number one. Here the spectrin, in the case of RBC, spectrin is the number one, is the slowest moving protein, and going through it, the globin is the fastest. Okay, then you can remember this chart, but I don't think it is that important. In this, spectrin is important, ankyrin, just remember the name. This anion exchange is important, just remember the name. Glycophorins is also important, just remember the name. Okay, this is what I told you earlier already. Yeah, this, there are some protein who are glycosylated, means like the glycogen glucose or glycogen residues are added on this membrane protein. This membrane protein can span over the lipid bilayer, which will called as the membrane bilayer protein, as the integral membrane proteins, while other will be at the surface, which will form the protein-protein interactions. Like here, ankyrin and spectrin are forming a protein-protein interaction. And then the RBC cell membrane contains an anion exchange protein and the glycophorins. Anion exchange protein is the band 3 protein, as we can see here. Anion exchange protein is the band 3 protein. These band 3 protein are the transmembrane glycoproteins. These are the transmembrane glycoprotein. Band 3 transmembrane proteins. These are the glycoproteins again. The carboxyl terminal of this band 3 protein will be outside of the RBC, and the NH3 or the amino terminal will be cytosolic phase, means inside the facing inside part of the RBC. The principal function of this thing is is to provide a channel through which the chloride and bicarbonate ions formed by the hydration of CO2 can be exchanged. Means what I'm trying to tell you, I'll explain it here. Let's say this is an RBC. There is chlorine inside of an RBC, and there is an CO2 at the tissue level. This CO2 in the form of bicarbonate will be taken up by the RBC, and the RBC in counter to that will give the chlorine to the tissues, or it will secrete in the membrane there. This is done by the channel or the protein which is known as the band 3 protein or the anion exchange protein. And similarly here, this process will be reversed at the lung level or the respiration level, where we are exchanging oxygen in response to the CO2. This CO2 will be going out, and the chlorine from the outside membrane will be coming in, so that RBC will have chlorine uh, that can be exchanged again at the peripheral tissue level. This band 3 protein, the amino terminal which were facing cytosol, which had a cytosolic phase or which was facing inside part of the RBC, will bind to the another proteins in the red blood cell, which includes the band 4.1 protein, 4.2 protein, ankyrin, hemoglobin, and several glycolytic enzymes. Then what are the glycophorins? There are three types of glycophorins: glycophorin A, B, and C, in which the glycophorin A is the major abundant form of glycophorin. It is compressed of the 131 amino acid, the polypeptide sequence, in which 16 covalently bonded oligosaccharides are present, and this oligosaccharide hydroxyl group is bounded by the serine and threonine residues. The hydroxyl group, here I'll try to show you. This is the structure of serine. Here, this is the main carbon or the only carbon of the serine, which is bound to the R, that is the alkyl groups or other polysaccharide residues or the oligosaccharide residues. And this is what is the hydrogen, and this is the what we need here is the hydroxyl group. This is the hydroxyl group, and which binds to these oligosaccharides and will form, and with this bounding we will form the glycophorin A, complete structure of the glycophorin A. Similarly, in the case of threonine, this is the structure of threonine. Instead of directly connecting R to this carbon, we'll just add in one methyl group, and this will termed as the threonine. Here is the hydroxyl group of the threonine, and where these covalent bond form, the oligosaccharides will be attached. Then the amino terminal of this, carboxyl terminal of this oligosaccharides or the glycophorins are extended into the cytosols, where they binds to the protein 4.1. Here we can see this for this point, that is the anion exchange protein, band 3 protein. This is the band 3 protein. See, it is binding to the another proteins of the RBC, that is the glycophorins, ankyrin, spectrin, and actin. Here we can see it's like backside of it. It will bind to the actin also, and protein 4.1 and other proteins also. In the case of what glycophorins, these glycophorins will bind to the this membranes, where the glycophorin, this glycophorins will bind to these band 3 proteins, band 4.1 proteins, and 4.2 proteins, which is not shown in that. Then genetic polymorphism is the case in which the RBC or the glycophorin A fail the glycosylation of the glycophorins, which is an important base in the MN blood grouping system. There are various blood grouping system, about 30 various blood grouping systems are present, in which the MN group system, ABO system, and the RH system are the three effective and the major systems of the blood grouping. But even if some person or the patient doesn't have this glycophorin A, there will be no adverse effect shown physiologically or the morphologically. But this glycophorin somehow contribute to the shape of an RBC, which is very very important for us and uh, can be distorted by some enzymes or not enzymes actually, some bacteria or the viruses. And the major another major thing is this glycophorin serves as the binding sites for the viruses and the bacteria and malarial parasites, like plasmodium falciparum. They at this point it may seem that not having glycophorin is better, because this glycophorin is providing a space or binding site for the viruses and pathogens, but it maintains the shape of RBC and it's important for us. Then the spectrin, ankyrin, and another peripheral membrane proteins, which helps to determine the shape and flexibility of the red blood cell. As we discussed this already, that shape of an RBC is the biconcave shape, which is maintained by some certain group of proteins that are known as the cytoskeletal protein. These are strong mesh like protein which forms the mesh kind of structure in the network. In that, the spectrin is the most abundant protein responsible for the shape of an RBC. The spectrin has its two component, the alpha component and the beta component, that's alpha chain and the beta chain. This alpha chain and beta chain intertwine in with each other and forms the dimer, and this dimer again goes and intertwine to form the heterotetramer. Dimer means this is alpha chain, this is beta chain, and it will be like wounded to each one of them, and it'll form, this is the dimer one, and another two dimer will form the heterotetramer. How? And this is an anti-parallel orientation, means we start one, like this is considered this as an alpha chain, this is going from, this is the tail, this is the head, it's going like this, but the another beta chain will come like this and bind like this, just same as structure as the DNA, double helical structure, but head to head anti-parallel orientation. The this spectrin molecule binds to the another protein or the components of the RBC to maintain the shape of an RBC and form the mesh kind of structure, that is the ankyrin actin band 4.1 protein. Yeah, this is what I told you already, that is strong enough, this forms maintains shape of an RBC. And this shape or the strong uh, network of these proteins makes RBC strong enough to not to burst or destroy because of the osmotic pressure produced by the failure of exchange of water and the sodium. Then another protein is there which also contributes to the shape of the RBC, that is the band 2 protein, also known as the ankyrin. It is a pyramidal shaped protein. The ankyrin is sensitive to proteolysis. It binds to the protein bands 2.2, 2.3, and 2.6, and all of these bands are derived from the ankyrin, intact ankyrin, means this is the ankyrin, and after some time this start to proteolyze or some start to go away and will form the another kinds of brands on the SDS page, and will term them as the 2.2, 2.3, 2.6, but all of them are formed from the band ankyrin or the intact ankyrin, and which collectively forms the band 2.1. Then there is another protein that is known as the actin, which is an band 5 protein. It is an globular. There are two forms of actin: globular actin and the F actin, that is the filamentous actin. Globular actin will eventually be converted to the filamentous actin, and this F actin is the monomer which rapidly polymerize and extend in the double helical microfilament, and this microfilament will bind to the spectrin and band 4.1, and will again form that strong network of this protein, the strong mesh of this protein, which will maintain the shape of RBCs. Then there are proteins, that is the band 4.1, which we are uh, you know I'm telling it from longer time since the start of this chapter. What is band 4.1 protein? Band 4.1 protein is the globular protein in shape, and it's tightly site near the actin binding domain in the spectrin, in the tail of spectrin, to form the protein 4.1 spectrin actin ternary complex. And this protein 4.1 is responsible for to the binding to the integral membrane of glycophorin A and glycophorin C. This is what I was talking before. This is how the glycophorin A or the glycophorins maintains the shape of an RBC. And then other proteins that, like, which include band 4.9, adenosine, and the tropomyosin, which also maintains the shape. Then there are some abnormalities. If there is something normal, means there will be something abnormal. It's not the word from the god, but yeah, there will be something abnormal. And the abnormalities in the spectrin causes the hereditary spherocytosis and elliptocytosis, which we discussed already very shortly. Here we'll go in little bit depth. That is, hereditary spherocytosis is genetic disease transmitted as an autosomal dominant disease. It's characterized by the hemolytic anemia. We have seen this already, and the splenomegaly will be the final destination of this kind of RBCs for the destruction. Then these spherocytes are the less deformable and more prone to destruction in the spleen. Means they are not that much deformed, but they are converted into much more globular shape and prone to the destruction in the spleen, which we saw already. Then this hereditary spherocytosis is much more common in the northern European ancestry, and which is caused by the amount or the deficiency in the amount or the abnormalities in the structure of spectrin, and less frequently, very very less frequently, in the ankyrin band 3 protein, band 4.1 or 4.2 proteins. Then this spectrin allows the erythrocytes to swell in the spherical shape. That means loss of spectrin or the impairment in the structure of spectrin allows the RBC to swell in the spherical form. And generally, this hereditary spherocytosis is relieved by the surgery, means splenectomy will remove the spleen of the patient. That means there will be no trapping of this RBCs in the capillaries and destruction by the macrophages. The hereditary elliptocytosis is almost same as the spherocytosis. The difference is that RBCs in the spherocytosis was spherical. In the elliptocytosis, they are elliptical, more in elliptical form. In spherocytosis, their occurrence of this spherocytosis was one patient in 5,000 another persons, which was more common in northern European descents also. And in the elliptocytosis, it's more common in like northern European descent is the same, but which is like the frequency of it is 1 is to 200 and 2,500 persons. Then this is the main cause. Yeah, hereditary elliptocytosis is mainly present where the malaria is common, because uh, as we see this mm, wait, okay, sorry for the break. Yeah, this elliptocytosis or the hereditary elliptocytosis is much more common where the malaria is present, and it's caused by the genetic abnormalities which affects the proteins, the membrane proteins which maintain the shape, that is the spectrin, less frequently band 4.1 and glycophorin C. Then the biochemical basis of the ABO system. Finally, in the some last part of this chapter, that is the ABO system or the blood grouping system. The term blood group is referred to as a defined set of red blood cell antigens and blood group substances controlled by the genetic locus having variable numbers of alleles. The term blood type refers to the antigenic phenotypes usually recognized by the appropriate antibodies. The summary of this passage or this paragraph is that whatever the blood group we have, for example, I have A positive, that is A. What is A? A is the phenotype, that is an antigen. It is an phenotype. It is an phenotype antigen in our blood. That means if I have A type of antigen, I will not have A type of antibodies, anti-A antibodies. I won't have anti-A antibodies. I'll have anti-B antibodies, and I will have, if I'm A positive, I will have sorry, I won't have any kind of antibodies. But if I'm negative, I will have anti-Rh antibodies. That's it. What How do we like calculate, or we figure out what is our blood group? This process is like conducted in various steps, or it is checked in parts. Like for A, the slide is different. For B is different. For positive negative, that is the RH, the slide is different. And we'll see the agglutination reactions, which is caused by the blood on that specific side. The process is, we get a slide, we coat it with the antibodies. That is, on first slide we'll coat with the anti-A antibodies, on the another anti-B antibodies, and the anti-RH antibodies. What will happen, and then we'll prick here or somewhere, we'll draw very small amount of blood, and we'll put one dots on each slide, or some amount of blood on each slide, and we'll wait. And then we'll see which particular slide forms the agglutination reaction. That means the clot formation we can like see. We don't even need microscope if we have ample amount of sample. Okay. If there is an agglutination reaction, that means that is an R blood group. uh, how can I explain it? Like, as like our slide is already coated with antigen. Okay. And then we put an blood on that, which contains a blood group, or the A antigen, and the antibody is B. Sorry, antibody is also A present on that slides. Means there will be an antibody antigen antibody reaction, and there will be a clot formation, or the destructions of RBC, or sorry, platelets in that in that slide, and will form the clot. But on the other slide, on the B slide, on the RH slide, if my blood group is A, I won't form any clot on B slide B. But if I'm A positive, I will form a clot on RH positive slide. And if I'm negative, I won't form a clot on RH slide. Okay, that's the simple thing. This is an all RH ABO blood grouping system. We'll just go fast forward through it. The ABO system is a crucial importance in a blood transfusion. It was developed by the Landsteiner in 1900. Okay, the individuals of the type A have the anti-B antibodies. We saw it already in their plasma, and that will agglutinate if the person is transfused as a receive a transfusion of the type B, then and then only. Not like type A is present and then anti-B body will attack and form the agglutination reaction. No. In the erythrocyte, type B will have the or AB will have the another kind of anti-A antibodies, and will form the agglutination reaction. But what is O? O means there is no antigen at all on the membrane of RBCs. We have only and only antibodies in the blood group O. Means there will be antigen antibody A, antigen and anti-A antibody, anti-B antibody, and the RH antibodies will be present if you are O positive, and if you are not, then the antibody won't be there. Okay. Then the type AB blood has neither anti or anti-B antibodies, and they are designated as the universal recipient. Blood O, we discussed, neither A or not B. They don't have any antigens. Then universe, and that's why they are universal donor. In that particular type O, O positive is not an universal donor. Type O is a universal donor, but not for the O negative blood groups. It is only for the positive blood groups like A positive, B positive, AB positive, for them only. But O negative is the one who is the universal donor. Who is the universal donor? Then there's further group are simplified as A1, A2, yeah, for the Mendelian things and allelic things and all. The thing here is to remember is this: uh, the substance or the chromosome responsible for the production of A, B, and O, or AB groupings is the chromosome 9, long arm of chromosome 9. Then the ABO antigens are glycosphingolipids or glycoproteins. The ABO antigen or as they are sometimes referred to as substance consisted of set of complex oligosaccharides. Means like how we determine how A becomes A, B becomes B, and O is the nothing. There are some sort of oligosaccharides present on the membrane which determines the A or B antigen of that particular RBC or R blood group. Then we have the covalent attachment on the membrane lipids, the forming type molecule known as the glycolipid. This is just to remember. In secretion, same oligosaccharides are anchored to the proteins, forming the glycoproteins. The presence in the secretion is determined by the gene designated as SC. This is important. The name of gene is SC or for secretor, which codes for the specific fucosyl transferase. Remember, this fucosyl transferase is the precursor of all type of blood group, that is A, B, and AB and O also. For O, it's the H. We call it as an H substance. In some cells, in other cells, okay, in individuals having SCSC cap dominant secretor gene, or one dominant and one recessive genotypes, will secrete either A type or B type antigens and will have that particular type of group. But what if we have both recessive types of genotypes? Then we won't be able to secrete any type of antigen. That means we will have an O type of blood. Red blood cells, both genotype express A and AB and globin. Okay, there is of nothing. Okay, then the gene A encodes for the GalNAc transferases, and the gene B represents or the codes for the Gal transferases. This GalNAc and the Gal transfer are the enzymes which are responsible for the production of A substance and the B substance. This is an H substance, which or the O substance in the case of O groups, because the O groups does not form any reaction and it does not have any kind of antigens on their red blood cells. The GalNAc transferases acts on the H substance to form the A substance, that is the blood group A, and in the B blood group, the Gal transfer, this enzyme acts on the H substance and will form the group B, that is the blood group B. That's very simple. It's complicated over here, and that's the summary of that. Then individuals of type AB will have both enzymes, and thus they synthesize two oligosaccharides chain. Means they will have oligosaccharide chain of A also and the B also. And then type O will harbor frameshift mutation and the gene in the gene encoding and terminal glycosyl transferase that result in the production of inactive protein. Thus, the H substance constitutes the O antigen. There is a frameshift in the case of type O. There will be a frameshift mutation in the encoding gene, that is the SC or the secretor gene on the long arm of chromosome number 9, and because of this mutation the protein will become inactive, and that's the reason why H substance acts as the or constitutes as the O antigen. Then an H allele arises from the mutation in the process of H locus, that is like here remember this H and this H, these are like dominant and the recessive things. In the case of heterozygous H genome type, they can still synthesize adequate level of H substance. Means that they can show either A type of blood group or the B type of blood group or AB type of blood group. But what if the case is recessive H or the homozygous recessive individuals? They cannot synthesize any levels of these protein or the substances and cannot show any type of antigen, since H substance is precursor for H A and B that we saw already. Then this HH or the recessive genotype will have red blood cells of O. That's now what I said. And then there is nothing. This Bombay phenotype is the, yeah, they express one of the both or the, wait. Yeah, this red blood cell will have the type O, regardless whether or not they express one of the both A or B terminal glycosyl transferases. Means if they show or they do not show the protein formation reactions or these reactions, the this O type will refer to as the Bombay phenotype, because they have an O. It's like H becomes an HH, like recessive H, recessive H forms the homozygous recessive genotype or the phenotype, and will replicate in as the type O blood group. Then the last part, that is the platelets, and let me drink a water. Thank you. Okay. Then platelets are also devoid of majority of its organelles, but some organelles are still present in the platelets, which are the mitochondria, lysozyme, and the tubular network, that is the open canonicular system. Here I will explain you the open canonicular system. This open canonicular system is like, let's say this the inner round or the circle is the our platelet, and this is its membrane or the outside membrane, and this tubular shape pores which are like coming inside of this platelet forms the canalicular open canalicular system. And the importance of this canalicular system is that the granules, the dense granules and the alpha granules which are present inside of this platelet, can go out when they are called on, means like when there is an signal from the outer environment. This granules will secrete, and the macrophage and platelets will act on that will act on that injury or that bruising site, and will form a clot. That's it. In this alpha these factors or the clotting factor are stored inside the platelets, which are densely packed in the two structures or the secretory vesicles called as the dense granules and the alpha granules. These dense granules contains calcium, adenosine triphosphate ADP, serotonin, while the alpha granules contains fibrinogen, fibronectin, platelet derived growth factor, and von Willebrand factor, which causes an von Willebrand disease, and the other coagulation factors ready for the release on the receipt of appropriate stimulus. Then, under the normal circumstances, this small diameter and enucleated cells circulate densely at the speed of 2 to, like they are present at the 2 to 4 into 10 raised to 5 platelets per millimeter in the blood, while the platelet derive majority of their energy by metabolizing the fatty acids through the process of beta oxidation, as they have mitochondria. Wow. But the erythrocytes or the RBCs, they do not have. So the major source for erythrocytes is glycolysis. Then disorders of the platelets or the platelet disorder compromises the hemostasis. The abnormalities in platelet number and function can have serious physiological consequences like acute coronary syndrome, that is the formation of large hyperreactive platelets result in the increased risk of thrombosis and also increases the risk of myocardial infarction. Then this is the myocardial infarction which increase by the acute coronary syndrome. Okay. Then another disorder of the platelet is the immune thrombocytopenic purpura. It's caused by the formation of antibodies against one's own platelets. Means our body will form the antibodies against of our body platelets itself, and this is an autoimmune disorder. The marker of this autoimmune disorder is the thrombocytopenia, means decreased quantity of the thrombocytes in the blood. When this platelet surface becomes decorated with antibodies, means let's say this is an platelet, this yellow site or the yellow shape I'll draw just for you. This is an our platelet, and these lines are our antibodies, means decorated with antibodies, and its antibodies are attached or attacking to trying to destroy our own platelet, and this will form a larger structure in a shape, and while passing through the spleen, this will be attacked there and will be uh, you know clarified by the splenic macrophages or the destroyed by the splenic macrophages. But in some cases, what happens is that this autoantibodies can bind directly to the megakaryocytes. Megakaryocytes is the mother cell or the platelet forming cell. If these antibodies, not antigens, antibodies binds to this megakaryocyte, that is like obvious. The platelet formation or the rate of platelet formation will be again reduced or decrease. Then this is the thing is known as the thrombocytopenia again, depressing the production. Thrombocytopenia can occur in the persons with the homozygous for a mutant variant of glycoprotein IIB or IIIA, which will leucine 33 is replaced by the protein. To explain this, let's come here. Let's say you are the one who is an homozygous mutant for this glycoprotein IIB or IIIA, whose leucine at the 33rd place is replaced by the proline, and you are this homozygous person and you receive a transfusion like blood transfusion from the another person same as you, that is the homozygous person or the heterozygous person, but which it is a dominant type of heterozygous person in the wild type. Majority of platelet majority of what happens, the major platelet antigen will this oh sorry this exposures of the donor's platelet will trigger the production of allo antibodies, means the your uh, body will form the antibodies against the platelets of the donors, means the blood which you receive from the another person, from the donor blood. Your body will produce antibodies for that platelets, which is known as allo antibodies. But these allo antibodies can attack your own platelets like formed by your own body, and this causes again the thrombocytopenia. And then in the neonatal alloimmune thrombocytopenia, which affects roughly 1 in 200 term pregnancies, what happens in the neonatal alloimmune thrombocytopenia is the this, let's say this is the mother, this is the uterus, and this is the fetus. From mother, the platelets from the mother, antibodies from the mother travels, not platelets, antibodies from the mother travels to the fetal fetal blood circulation through the placenta, and will destroy the fetus plates of the fetus in fetus, and which will again make the fetus thrombocytopenic. Then the thrombocytopenia can also be caused by the drugs such as tamoxifen, ibuprofen, which we prescribe in some conditions like rashes and fever, vancomycin, and many sulfonamides. To summarize this passage, I'll just come over again. The first thing, there's the immune thrombocytopenic purpura, which is caused by the antibodies, which antibodies these antibodies are destructing your own platelets. Then the second thing is that these antibodies can directly attack the megakaryocyte, which is a mother cell of the platelet formation, which will decrease the platelet number and which will slow down the rate of platelet formation, which will form the thrombocytopenia again. The third condition is that homozygous mutation in the glycoprotein IIB or IIA, where if the person receives from the another homozygous or heterozygous wild type anti platelet antigen, which will produce the antibodies for that donor's blood, and these antibodies are known as allo antibodies, and these antibodies can attack your own or the person's own platelets. The neonatal alloimmune thrombocytopenia is the mother's antibodies will cross the placental barrier and will attack the fetal platelets and destroy the fetus platelets, and this thrombocytopenia can be caused by the drugs such as tamoxifen, ibuprofen, vancomycin, and sulfamides. In the symptoms of the hemolytic uremic syndrome, it's a disease in infant, means the babies who are from the birth to up to the 1 year of age. 1 year of age are known as the infants, and this is mainly present in this infant category of the babies. A disease of infant characterized by the progressive kidney failure. These babies have the mm, down kidney function or the chronic kidney failure or the kidney failure, which means are or not are the infant's kidney won't be able to produce enough amount of erythropoietin and the thrombopoietin. Both enzymes are produced by the kidneys. That means if the thrombopoietin is not present, that means there will be a decreased production of platelets, and which will again cause the thrombocytopenia. Simple. By contrast, there is a sometime an abnormal bleeding caused in such in a case of von Willebrand disease. It is a genetic defect which compromises the ability of the platelets to adhere and aggregate. Means, let's say this is an our skin and there is some bruising or some cut on this our skin or some tissue or somewhere in not in the inside the body, out only. If it forms inside, it's very dangerous, far dangerous. It will clot formation inside body is very dangerous. Okay, if we have a cut on the skin, right, and then if have a cut, this platelet will be activated and will come here and form a clot. But in the case of von Willebrand disease, Bernard Solier syndrome, and the Glasman thrombocythemia, there will be a clot, but this clot won't be that strong enough which can resist the blood to go out of that bruise or the cut, and which will cause the bleeding again. Means the plate number of platelets is not decreased, but the function of the platelet or the aggregation of platelet is decreased. It's the same. All three of these are genetically inherited diseases. Then, recombinant DNA technology had an profound effect on hematology. In the cases of thalassemia or the many disorder of coagulation, we have greatly been benefited by or clarified by the investigation of utilizing the gene cloning or the DNA sequencing. Uh, then this conditions like this recombinant gene technique can uh, help us to understand more about the leukemia and the chromosomal translocations and advancement in the case of leukemia. This recombinant DNA technology was very useful in the case of production of erythropoietin, which we have to administer if the patient cannot produce more like normal amount of RBC or the patient in short is anemic. Uh, this recombinant DNA technology is very successful in growth factor synthesis also. And the first pathophysiology condition treated by the gene therapy was the deficiency of the adenosine deaminase. This technique was developed by the uh, Dr. William French Anderson in the 1990. It is introduced by the new copy of gene carried on retroviral and 4-year old. So this is just normal information. Yeah, this is the summary of chapter which you can read later. This is not a perfect summary of chapter. There are many more points from for which you have to read chapter or we have to read chapter. More importantly, these are references, and thank you. Subscribe to my channel and follow for more videos. Thank you guys. Bye bye and see you again in the next video.